Now,
Yes, sir, visible.
just we have finished drug regulatory affair introduction. So what actually RA expert responsibilities are there with respect to product life cycle. Now we will see regulatory requirements for drug approval. Okay, in detail. So regulatory requirements for drug approval, why it is needed? We should have some reasons. Otherwise, anyone can manufacture their own product and directly sell to market. Is this possible with respect to drug product? No. Reason behind this, we have various reasons why we should go for regulatory requirements specific to specific countries. So it is required. Why? We have different reasons. We will discuss that in this chapter. Okay. We have two examples why we need to go for specific regulations for new drug approval. So there is thalidomide tragedy. I think we have discussed that part before also. In 1962, there was one incidence, that is cocomelia, that means malformation of limbs. So in that particular period, some of pregnant ladies were taken thalidomide drug. Okay, and because of that thalidomide drug, in that particular 1-2 year, it is observed that there is issues with different organs of offsprings. Okay, so after delivery, it is observed that some childs have no limbs, some have no ear, no finger. Likewise, it is observed in 1962. So around 10,000 plus cases were reported, and from that 50% get survived. Okay, 50% get survived. That means 50% deaths are there because of thalidomide in 1962. There is one issue, that is teratogenic effect. Okay, before that we are not going for teratogenic testing. Okay, that means once drug is in market, before drug is in market, we need to go for teratogenic study also, so that we can get the information about whether that particular drug is having some effects on pregnancy. We need to check that. And there is one example also, elixir sulfonamide in 1937. So here also there is one solvent, that is diethylene glycol, which act as toxin. Because of that also around 100 plus deaths were reported in 1937. If you see this type of incidents, in that case we can think like there should be a system so that we can get a safer drugs. Okay, there should be a system, there should be some for developing new drugs, for marketing different formulations in society. And because of that only there is introduction of regulations for new drug approval. Because of this type of incidences, drug approval process gets started. That is general flow chart here discussed. Drug discovery, that is first step for any type of drug, then drug development and manufacturing. So in that we'll have some clinical, non-clinical studies. Once it is get approved, we can go for clinical trials in humans, then marketing application. During these phases, we have different study protocols, and we need to get approval from different regulatory agencies. So in different stages, we need to file different documents to the regulatory agencies. Once they find it is okay, once they find it is safer for human being, then they will approve that particular drug for use in society. Currently, different countries follow different regulation requirements for approval of drug. New drug application. So that is once drug having its preclinical as well as clinical study reports and they are safer for use. In that case, we can file new drug application to the regulatory agencies and they will approve the same if it is found that it is okay for human use for specific therapeutic application. So NDA is an application submitted preclinical and clinical test data for analyzing the drug information and description of manufacturing procedures. So that means what? NDA application, it includes preclinical as well as clinical data. Okay. And where in that particular document, it is explained how that particular drug is safer for human application. Once it is submitted to the regulatory authorities, they will either approve the same or they will say it is approvable. We need some changes in that particular, or we need additional documents. Then it can be approved. And third consequences is not approvable. So regulatory agency will check whether that particular product data is okay or not. If it is okay, then they will give approval to the particular manufacturing company. If it is approvable, that means it the results are okay, but at some extent. In that case, we can get some additional documents. We can go for some additional studies so that it will get approved. And third consequences will be it is not approvable. That means product which is studied in that particular phase will not be approved by regulatory agencies. So here for different processes in drug development, we have different regulatory agencies checking all the activities carried out by different pharmaceutical companies. We should have specific team so that we can go smoothly. Drug development is the process of bringing new pharmaceutical product or drug to the market once a lead compound has been identified through process of drug discovery. So that is what a basic statement is. The process of drug discovery and development is very long and need around 10 to 12 years. That we have already discussed that part. Means a single molecule come in market, it require around 10 to 15 years, which includes the close interaction of large number of scientific disciplines. So in that, lot of money get invested, manpower is required, facilities required. Okay, the team who is working there, they have their own responsibilities. They have their own responsibilities. That means drug development team is responsible for planning research studies to further characterization of drug candidate. So they will check the report which is submitted to the regulatory agencies so that they can go for further characterization of the drug. They can integrate new research results with previously generated data. They can compare preparation of detailed drug development plan. That is also work of drug development team. Then reviewing research result from experiment conducted by various scientific disciplines. Monitoring the status of ongoing research studies. That means whatever they have approved various research projects, in that case they will monitor whether that particular stages are achieved within that particular period or not. They will check if there is need of modification, if there is some addition of data, in that case they can modify the plan also. And last one is comparing research results and development status of drug molecules of competitors. So that is work of drug development team. That is most important. The new drug approval is of two phases. The first phase is clinical trials, and second one is phase for marketing authorization of drug. So these are two stages. We will discuss that in detail. Firstly, non-clinical studies of drug are completed to ensure efficacy and safety. So non-clinical study मध्ये आपण काय करतो? We need to check whether that particular drug having efficacy and safety, and then application for conduct of clinical trial is submitted to the competent authority. Once it is observed that particular drug is efficacious and safe, we can go for clinical trials. Okay, so safety and efficacy, once we have access, in that case we can go for investigational new drug application. We submit IND application to the regulatory authority. Once we get approval, then we can go for clinical trials. Okay, clinical trials is submitted to the competent authority of the concerned country. Thereafter, the clinical trial can be conducted phase 1 to phase 4. So we have different phases in clinical trial, phase 1, 2, 3, 4. We have difference in volunteers that we will discuss in detail also in this part. The studies are performed to ensure efficacy, safety, and optimizing drug dose in human beings. So that is first part. Okay, in phase 1 trial, generally we go for efficacy, safety, and dose optimization. After the completion of clinical studies of drug, then the application to the competent authority of the concerned country for the approval of drug for marketing is submitted. So that is general procedure. The competent authority review the application and approve the drug for marketing only if it is found to be safe and effective. So that is most important. Drug should be safe and effective in human beings, or drug have more desirable effects as compared to other adverse effects. Even after the approval of new drug, government should monitor its safety due to appearance of some side effects. Okay, post marketing surveillance मध्ये आपण बघतो, we need to check the compliance of that particular product in marketing page also. When it is used in larger population, the interaction with other drug which were not assessed in a pre-marketing research trial and its adverse effect. So after post-marketing surveillance, we can say during that page, we need to check effect of that particular drug when given singly or in combination with different drugs. In that case, we need to check adverse reactions of that particular drug also. Non-clinical drug development, that means preclinical study. Before clinical study, we go for preclinical studies. Okay, a laboratory test for novel drug or new medical device is usually done on animal subjects. So generally, we do here animal studies to see whether that particular product is active or not. We have three types of studies in that preclinical studies: in silico, in vivo, and in vitro. In silico, that means computer simulation. Computer simulation, that means by different softwares, computer aided drug design जसं म्हणतो आपण, तशा पद्धतीने इकडे सुद्धा आपण animal study predict करू शकतो. That is computer simulation. In vivo म्हणजे within a living, that means animal study. We can use rats, mice likewise, or horse, rabbit. In vitro, within the glass, outside the living organism. These are three different types that comes under preclinical trials. This process of non-clinical development of medicine is very complex, time-consuming, and regulatory driven. Regulatory driven, that means whatever the things we are carrying out in that particular company, it should be regulatory driven. That means all the regulatory guidelines, procedures must be followed by that particular organization. The primary aim of non-clinical development phase is to analyze and determine which candidate has the greatest probability of success assess its safety, and raise firm scientific foundation before transition to the clinical development phase. So here, generally we check here in this page, we have number of molecules in our hand. Okay, we are checking its safety, we are checking its clinical performance, we are checking its related issues in that particular phase. Okay, pharmacology, study of effects of chemical substances on living systems. So here in this non-clinical study, it holds all the aspects of drug discovery, ranging from details of interaction between drug molecule and its target to consequences of placing the drug in market. So we need to study all that under pharmacology part. Okay. Selectivity testing, that means what? It consists of two main stages: screening for selectivity and binding assay. Screening for selectivity in the sense, in pharmacology we know there is drug receptor interaction to have a pharmacological action. In that case, we need to check whether that particular compound is binding with specific receptors. We need to check that, and binding assay. This is second part. To determine the potency of drug, the selectivity of compound for chosen molecule targets need to be assessed. So that type of studies are carried out under selectivity testing. And pharmacological profiling means what? This include determination of pharmacodynamic effects of new compound either in vitro or in vivo models. So pharmacodynamic effect, that means what? Yes, pharmacokinetics. In pharmacokinetics, we study ADME profile of drug, right? And in case of pharmacodynamics, we check the effects of drug on human body. Okay, mechanism of action and all that will be studied in pharmacological profiling. The next part is safety pharmacology. Safety pharmacology means apart from therapeutic effect of particular compound, we need to check safety related issues particular to that product. This includes the scientific evaluation and study of potentially life-threatening pharmacological effects of potential drug, which is unrelated to desired therapeutic effect, right? So apart from therapeutic effect, we are checking potential life-threatening pharmacological effects. That means we are checking by increasing or decreasing the doses. Okay, which type of effects are observed. That is most important. These tests are conducted at doses not too much in excess of the intended clinical dose. Safety pharmacology seeks to identify anticipated effects of new drug on major organ function. So that is most important. We are checking the specific effects on different organs of our body. It is aimed at detecting possible desirable or dangerous effects of exposure of drug in therapeutic doses. So we are checking by changing doses. Toxicological approaches to drug discovery, acute and chronic. So here in this acute toxicity studies मध्ये आपण काय करतो? Acute toxicity studies are generally carried out on mice and rats. Okay, using the same route as intended for humans. In addition, at least two or more routes should be used to ensure systematic absorption of drug. Here, we are carrying studies on mice and rat, and we are considering two different routes. Okay, used to ensure systemic absorption of drug. This route may depend on nature of drug. So, based on type of drug, we can select the route of dose administration. Then mortality should be looked for the 72 hours after parenteral administration and 7 days for oral administration. We need to check the effects of drug. We need to check mortality rate when that particular animal, mice or rat, got killed. Parenteral administration, we have 72 hours we need to check, and for oral administration we need to check for 7 days. The symptom, signs, mode of death should be reported with appropriate macroscopic and microscopic findings where necessary. We need to submit this data also to the regulatory agencies. So that is comes under acute toxicity study. Okay, we are doing studies for new chemical entities in this way. Long term study, these studies should be carried out in at least two mammalian species, and out of these two mammalian species, one should be non-rodent category. Okay, non-rodent म्हणजे rat, mice categories असतो नसावा. The duration of study will depends on type of factor. Means what type of studies we are doing, it will depends on that. Means we have different reasons that post marketing permission असेल, we have clinical trials or different phases in trials. Based on that, we have different duration of study. If a species is known to metabolize the drug in the same way as humans, it should be prepared in long-term toxicity studies. So ideal candidates for long-term toxicity study is if the species is known to metabolize the drug in the same way as that of humans. In that case, we can go for long-term toxicity study. The drug should be administered 7 days a week by route intended for clinical study in humans. So this is general procedure in long-term studies. We have different regulatory agencies. I think we have already discussed in regulatory affairs section. US FDA, that is for US, CDSCO, that is for India, EMEA, that is for European Union, Minister of Health for Sri Lanka, and DDA for Nepal. So these are different regulatory agencies. They are controlling all the aspects related with new drug product life cycle. Different types of drug applications that can be submitted to FDA. Once we know that particular chemical entity is having specific therapeutic effect, in that case, we start drug discovery. Once we assess the efficacy, safety of that particular chemical compound, we can go for investigational new drug application. So these are different types of application we can file to the Food and Drug Administration of that particular country. So IND means investigational new drug application. NDA means new drug application. So what is difference between this? IND, if safety and efficacy study and dose optimization is once done, we can go for IND application to the regulatory agency. Okay. Then we can go for phase 1, 2, 3, 4 trial in NDA application. Okay, NDA, abbreviated new drug application, that is related to generic drug that we will discuss in detail. BLA, that means biologic license applications. FDA is reversible, responsible for protecting and promoting public health. The IND application should provide high quality preclinical data to justify the testing of drug in humans. Almost 85% of drugs are subjected to clinical trial for which IND applications are filed. Okay, we have the chart for that. Okay, if you see here, preclinical phase, clinical phase. In clinical phase, we have phase 1, 2, 3. Once we complete the phase 1, 2, 3 study, we can file NDA application, and after that we have phase 4 study. Okay, so general time course is given, purpose and subject is given. So generally at preclinical stage, we are doing studies at laboratory level on different animals. Here in preclinical phase, we are assessing safety and biological activity of drug. Generally, it requires around 1 to 2 years. Okay. The specific chemical entity, we need to check safety of that particular chemical component, biological activity. It requires around 1 to 2 years. And new drug pass, that means in this case, around 100% drug get passed. Okay. Then we file investigational new drug application. That is in pink color. You can see. Okay. Then we'll file investigational new drug application. Once it is approved, Okay, regulatory agencies will check whether that particular drug, whether that chemical compound is safer, whether that particular chemical compound is having therapeutic activity or not. They will check that reports. Okay, here also regulatory affair expert is important. He will compile all the data that is carried out in that particular 1 to 2 year. Okay, he will compile and he will submit to the regulatory agency, ज्याला आपण इन्व्हेस्टिगेशनल न्यू ड्रग ऍप्लिकेशन म्हणतो. ओके, याच्यामध्ये फक्त आपण सेफ्टी आणि बायोलॉजिकल ऍक्टिव्हिटी रिलेटेड स्टडीज चा रिपोर्ट असेल तो सबमिट करतो रेग्युलेटरी एजन्सीला, दॅट इज इन्व्हेस्टिगेशनल न्यू ड्रग ऍप्लिकेशन. once it is fast, once it is approved, then we can get permission for further studies, that is clinical studies. त्याला आपण क्लिनिकल फेज म्हणतो. इन दॅट वी हॅव 4 different stages: phase 1, phase 2, phase 3 and phase 4. So here in case of phase 1 we have 20 to 100 volunteers. Samples किती असतात आपल्याकडे? Around 20 to 100 volunteers are used. Here again safety and dose optimization studies are carried out. Okay, safety and dose optimization studies are carried out and it require around 3 to 4 years. This is general. It require around 3 to 4 years and around 70% of investigational new drug application get passed. Okay, समजलं का? हे प्री क्लिनिकल स्टडीज मध्ये 100% सगळे पास होतात, कारण याच्यामध्ये आपण सेफ्टी आणि बायोलॉजिकल ऍक्टिव्हिटी बघतो. पण फेज 1 स्टडी मध्ये आपण काय करतोय? वी आर चेकिंग सेफ्टी अँड डोस ऑप्टिमायझेशन. ओके, and it require around 3 to 4 years. So within that 3 to 4 year we are checking the effects on 20 to 100 volunteers. That is phase 1 study. In this case, around 70% of investigational new drug applications get passed. Then we have second phase, that is phase 2. Here in this case we are using around 100 to 300 patients for study. Here we are checking effectiveness of that particular chemical compound and side effects. Okay, it also requires 4 to 5 years to complete the study, and around 33% of IND applications get passed through this. So out of that 70%, around 33 get passed to the phase 3. So in phase 3, the quantity of patient samples are more than 1000. So generally 1000 to 3000 patients are considered for phase 3 study. Also it requires 6 to 8 years. Okay, so it requires 6 to 8 years. And here also we are verifying the effectiveness, long term side effects of drug. Okay, long term side effects of drug we are checking in phase 3. It requires around 6 to 8 years, and around 27% of molecules get passed in phase 3. Once it is found that the particular chemical compound is effective and having less or no side effects, in that case we can file NDA, new drug application. Okay, पहिला क्रायटेरिया काय आहे? फक्त सेफ्टी आणि बायोलॉजिकल ऍक्टिव्हिटी केल्यानंतर आपण IND ऍप्लिकेशन फाईल करतो, आणि फेज 1 to 3 स्टडी कंप्लीट केल्यानंतर आपण न्यू ड्रग ऍप्लिकेशन फाईल करतो. फेज 4 स्टडी इथं केलेला नाही अजून. Here once we got approval from regulatory agency, NDA अप्रूव्हल भेटल्यानंतर आपण फेज 4 ट्रायल करतो. फेज 4 ट्रायल म्हणजे थोडक्यात प्रोडक्ट मार्केटमध्ये ओपन झाल्यानंतर त्याचे मार्केटमध्ये जे काही कॉन्सिक्वेन्सेस रिलेटेड साइड इफेक्ट्स असतील ते आपण स्टडी करतो. दॅट इज फेज 4 ट्रायल. दॅट इज आफ्टर NDA अप्रूव्हल. ओके, सो दिस इज जनरल पाथ फॉर new drug. Investigational new drug application means what? We have already seen that we need to check safety and efficacy. If it is okay, then we can go for investigational new drug application. Once it is approved, we can go for clinical trials, phase 1, 2, 3, 4. It is application filed to FDA in order to start clinical trials. Once we confirm that we need to go for clinical trial. In that case, we need to file INDA application in humans. In the drug was found to be safe from report of preclinical trials. That means once it is confirmed that particular chemical entity is having effectiveness as well as safety in humans, in that case we can file INDA application. A pre-IND meeting can be arranged with FDA to discuss number of issues. So here, in this case also, before IND application, we can have pre-IND meeting with FDA to discuss design of animal research, protocol for conducting clinical trials, review the chemistry, manufacturing and control of the investigational drug. So याच्यामध्ये आपण जो काही ॲनिमल स्टडी केलेला असेल त्याच्या रिलेटेड इश्यूज, त्यानंतर प्रोटोकॉल फॉर कंडक्टिंग द क्लिनिकल ट्रायल्स, तो फ्युचरमध्ये जो स्टडी करणार आहे क्लिनिकल स्टडी, त्याच्यामध्ये त्याचा प्रोटोकॉल वगैरे असेल तो डिस्कस केला जाईल आणि रिव्ह्यू द केमिस्ट्री, मॅन्युफॅक्चरिंग अँड कंट्रोल ऑफ इन्व्हेस्टिगेशनल ड्रग. सो that is from preclinical stage we need to check and discuss in